Welcome to my presentation on polypeptide synthesis. My name is Charisma, and we will first begin by talking about the use of models in biology. Models in biology play a key role in simplifying and explaining biological processes that may be difficult to understand. They come in a variety of forms, as shown in this chart, to assist in visualizing abstract functions which allow scientists to make predictions. They assist students and scientists to create abstract concepts by providing visual representations that can be tangible and easy to analyze to make a conclusion on one's research topic. We will first begin by talking about the strengths of the use of models. Models represent biological phenomena that can't be seen by the human eye and replace the need for direct experimentation on living organisms, particularly when experiments can cause harm or distress. Models enable scientists to visually see these phenomena in a simplified or magnified form, fostering a deeper understanding of their function and interactions. However, models can usually have a downside to their benefits, as models can usually oversimplify complex systems, missing details that could be important, leading researchers to create incomplete conclusions. They typically rely on assumptions that may not always be true if the data is limited or flawed. Additionally, models can often lead to misinterpretation, as models do not adapt well to changes or new information. Let's move on to transcription and translation. So transcription is the process where the genetic information from DNA is copied into a molecule of messenger RNA. This occurs in the nucleus and is the first step which allows the genetic code to be used to build proteins. Translation is the process where the genetic information from messenger RNA is used to build a protein. This occurs in the ribosome, which creates a chain of amino acids to fold into a working protein. These steps talk about the process of transcription and translation. However, I will not be explaining it as it is shown in the video I will be providing later on in this presentation.
Okay, let's move on. I tried to pay, he begged me to stay. Babe, I'm not staying, I just wanna play. In the party.
So this is the example of a model representing the first step in gene expression. Transcription copies the information in a gene used to create a functional protein. Its goal is to produce the RNA copy of a gene's DNA sequence. For protein coding genes, this RNA copy or transcript carries the instructions needed to build a polypeptide. Enzyme driving transcription is RNA polymerase, which synthesizes a complementary RNA strand using a single-stranded DNA template. RNA polymerase assembles the RNA strand in the 5 to 3 direction, adding nucleotides to the 3 end as it progresses. As you can see, the helicase and RNA polymerase are moving together along the DNA strand. Transcription occurs in 3 stages: initiation, elongation, and termination. Let's have a closer look at each. Elongation, the second step, the RNA polymerase reads the template strand of DNA, synthesizing an RNA molecule by adding complementary nucleotides. The RNA strand grows from 5 to 3 and mirrors the sequence of the coding strand of DNA, except uracil replaces thymine. Taking a closer look at what this looks like. In the last step, termination, when RNA polymerase reaches a sequence called the terminator, it signals the end of transcription. The completed RNA transcript detaches, ready for further processing and eventual use in protein synthesis.
She was a fairy.
The RNA polymerase finishes transcription and the RNA strand is released. The DNA strands closes back together. This happens as the RNA polymerase detaches from the DNA at the terminated sequence and the double helix reforms, like this. This entire process is crucial because it sets the stage for translation, where the RNA transcript is used to build the proteins essential for life, and this is the final model for transcription, and we'll move on to translation.
Hey, come here, look at me. Hey, look at me. What did I say? No, come here.
So the first step of translation is initiation, where the mRNA will exit the nucleus and enter the cytoplasm, where the ribosome and the mRNA will bind together. So the mRNA contains a DNA base triplet, which can include adenine, uracil, guanine, and cytosine. The tRNA reads the start signal on the mRNA, which has the anticodon, the start codon, and the tRNA, and which in this case would be the AUG, and the tRNA brings the first amino acid to begin building the protein. So the tRNA reads the first DNA base triplet, which begins making the amino acid, which in this case would be MET. This is a continuing process as the ribosome reads the mRNA one step at a time, and tRNA molecules bring the correct amino acids to build the protein chain. These amino acids are linked together like beads on a string. As we can see, LIA and MET are connected together. And this entire process is known as elongation. So this process continues until it reaches the stop codon. So the ribosome reaches a stop signal in the mRNA, which in this case will be UAC, telling it that the process is complete. This makes TYR in the amino acid chain. The amino acid chain detaches itself from the tRNA. The protein is finished, released, and ready to do its job in the body. The result is a polypeptide chain that folds into a functional protein. So after the process of initiation, elongation, and termination, the amino acid chain folds into a functional protein, and in the end result, we get a protein like this. Thanks. So this concludes the entire process of transcription and translation. Overall, building a functional protein. Polypeptide synthesis is a vital biological process that produces polypeptides when amino acids are joined together by peptide bonds, leading to the creation of proteins. Polypeptide synthesis is essential for producing proteins, which are vital for structural support, enzyme activity, and immune defense in organisms. These proteins enable cells to function, grow, and repair. Proteins created through this process have an important role by binding with other molecules to carry out their jobs, meaning that shape and chemical formulation are very crucial. It provides a way for inherited qualities to be carried in DNA and mobilized in cell function. They act like enzymes to sustain life and play a central role in biology through their role in medical and biotechnological applications. However, any errors that occur in this process during translation and mutations in DNA can lead to long-term diseases like cancer. In mRNA, it consists of the direct product of transcription and carries the genetic code from the DNA in the nucleus to the ribosome in the cytoplasm. mRNA provides the template for assembling amino acids in the correct sequence and dictates the order amino acids are added to the growing polypeptide chain. Without mRNA, the genetic information from the DNA could not be transferred to the ribosome, as it would have no instructions to assemble amino acids into a specific protein. mRNA provides the sequence of codons that determine the order of amino acids, and without it, the process cannot start. In tRNA, it transports amino acids to the ribosomes and ensures they are incorporated in the correct sequence by matching the anticodon to the tRNA codon. It acts like an adapter that translates the genetic information from mRNA into a sequence of amino acids. Each tRNA molecule has a specific anticodon that pairs with a complementary codon on the mRNA strand. The ribosome creates the formation of peptide bonds between the amino acids carried by the tRNAs, linking them together to form a continuous peptide. This is essential for decoding the genetic information and building proteins for survival.
You didn't have to cut me off.